Welcome to this talk on the evolution of imperfection. I'm Lawrence Hurst, the Professor of Evolutionary Genetics here at the University of Bath. This talk is based in no small part around my book, which is called The Evolution of Imperfection, and you can find it at all the usual places. What I'd like to do to start with is ask you to do a thought experiment. Think of the word evolution and ask yourself what image comes to your mind. And I'd argue you'd be pretty unusual if it wasn't something like this. This image is, in many regards, a sort of canonical view that we have of evolution, running left, a sort of chimp-like organism, to right, modern human. And it's sometimes known as the March of Progress. Now, this is not an image that evolutionary biologists like especially, and that's because it has a lot of biases in it. So one is just a simple view that evolution is somehow a progressive process. That striding left to right is no accident. The other is that it is actually directional, that somehow it is leading somewhere. And most importantly, where is it leading? The idea is that humans are somehow the pinnacle of evolution, the end point of the process, the fairy on top of evolution's Christmas tree, you might say. And in fact, Shakespeare got there earlier in many regards. So in Hamlet, you'll find this great quote from Hamlet. "What a piece of work is man," he says, and concludes with, "We are the paragon of animals, the very best the animal kingdom has to offer." So there you have it, an evolutionary view which matches Shakespeare, that somehow humans are the end product of evolution, the very best. And actually, there's a good reason to suppose that actual evolutionary processes could be progressive. And that's because when we usually think about evolution, Darwinian evolution, we think that one type replaces another type. The one that's better suited to the environment is the one that wins out overall. And under that model, present-day species should thus be better adapted than prior species. And we sometimes call this survival of the fittest. And there's good reason to suppose that some of the time this is what actually goes on. This lovely moth here is the pepper moth, and it is in many regards our paradigmatic example of the evolutionary process. In the 19th century, in the UK, where trees went black because they were covered in soot, a different version of the same species of moth, one that was only black, rose up in frequency because they were camouflaged against the black background. Nowadays, if you look in Britain, you'll see this version much more commonly. This is a sort of whitey version, and that's because with the introduction of the Clean Air Act in the latter part of the 20th century, the trees all went back from being black to a much more mottled colour, and this moth is now camouflaged, so it went from rare to common instead. And so yes, species adapt to their environment. The one that best fits in is the one that increases in frequency, increases in number, and that's the one that we see predominating. That's the evolutionary process. But what I would argue is that in the last 20 or so years, we've seen a revolution in our ability to study DNA, the source of all evolution in many regards, and that what we've learnt from this challenges this progressive view of evolution. Now, before I get on to how it's challenged, let's quick look at what DNA actually is. So it is the molecule of inheritance. So why do identical twins look like each other? They have the same DNA that they got from their mum and dad. And likewise, why do sibs look like each other? Why do offspring often look like their parents? It's all down to DNA. So our DNA is in 23 chromosomes, 23 that we get from mum, 23 that we get from dad, so 46 in total. And each of those chromosomes, that's one long molecule of DNA. But when you break it down, it's made up of what we call base pairs. And you can think of these as a bit like the rungs of a twisted ladder. So you see there in the image, if you were to untwist that, you just get a ladder with rungs across it. Each of those rungs is a nucleotide base pair. And they come in 4 flavours, A, C, T, and G. And if one half of a rung is A, the other half of the rung will be T. If one is C, the other will be G. And when we talk about DNA sequencing, when we sequence something, we're actually just working out the order of all those A, C, Ts, and Gs just on one half of the ladder, as it were. Now, that used to be an extraordinarily expensive thing to do. So the first human genome, complete all of those 23 chromosome sequence, came out in about 2003, and it cost $3 billion. That's about $1 per base pair. What you see there in the image is just a random load of DNA with all those base pairs just simply lined up. And the whole project as a whole took 13 years to do. Now, remarkably, we can sequence a complete human genome for orders of about $100 or so. And so that, in terms of the price per base pair, much, much lower. And in time-wise, it also is incredibly fast as well. You can do it in real time, in about an afternoon or so. So with all this information about DNA, not just our DNA, but we sequence up lots of DNA from humans, but also lots of our animal relatives, but plants, animals, everything we can now sequence. And when we look at this, so here are indeed our 23 chromosomes, when we look at this, perfection is not the first thing that comes to mind. And in fact, I would say that our DNA leaves us with a whole series of real head-scratching enigmas, things we really don't understand. So before we get there, what is DNA actually doing? So I've told you it's made up of these 4 bases. It's very, very long. We've got 3 billion of these base pairs. But DNA is not the doing molecule. DNA is the inheritance molecule. But to actually do anything, it does something else. It makes something called RNA, and RNA makes protein. Now, protein is the enzymes that digest your food, for example. They're proteins. The muscle that enables you to move, that's protein. Just about everything we do is protein. So DNA is converted into RNA, and RNA is translated into protein. And the bits of our DNA that say, make this little bit of RNA, which gets converted to protein, those are our genes. So the first surprise is we don't have many of them. So if we look across all of life, you'll see that the number of genes that organisms have, so the little bits of DNA that make an RNA that make a protein, how many different ones do organisms have? Well, right at the bottom there, you see some bacteria. They've got a couple of thousand genes. The first red point, that would be a yeast, so a simple single-cell organism, but like us, it has a nucleus. Bacteria don't. It's got about 6,000 genes. And then moving on up, you go to the record currently is held by a single-cell organism, which has about 100,000 genes, but we find plants have loads, so wheat has got an awfully large number, 90,000 or so. And we find tiny little water fleas, for example, they've got about 34,000 genes or so. And our millimetre-long laboratory worm, it's got about 20,000 genes. So how many do we have? Well, you'd have thought we're slightly more complicated than a single-cell organism that's got shy of 100,000. We're surely more complicated than wheat and rice, aren't we? But here came the first mystery then. It turns out we've only got about 20,000 genes. So we do not sit high up on this chart at all. So there's our first mystery. And related to that first mystery is a second mystery. Well, what proportion of all of our DNA is actually making those proteins? Because it could be we just make very big proteins, of course, and so it's a lot of our DNA. But no, it turns out it's about 1.2% of all our DNA is doing that job of DNA makes RNA, makes protein. So not very much at all. Well, what's all the rest doing? So that exons of protein coding genes there in the bottom left, as it were, that's the 1.2%. That's the bit that does DNA, makes RNA, makes protein. Most of the rest seems to be dead or dying jumping genes. Our DNA is full of bits of DNA that can just simply copy themselves. And most of it seems to be like that. And lots of the other stuff doesn't seem to have much of a utility at all, at least not obviously so. Now, you might think, well, maybe that's just the way DNA is. But no. So you can ask, well, how much DNA sits between our genes? And we also have stuff within our genes that we don't need as well. That has to get snipped out. These are things called introns. And it turns out that when we look across different species' DNA, so that's what you're seeing in the left-hand plot there, if you've got a lot of stuff between genes, you've got a lot of rather redundant stuff within genes. And as you can see, we're sort of right at the top of what we call genomic bloating. We seem to have lots of DNA we don't obviously need. But it's not at all inevitable. Lots of other organisms aren't like that at all. So we have an enigmatic load of DNA. We haven't got many genes, a quarter of that of wheat, as we saw. And very little of our DNA seems to do the canonical job that DNA is supposed to do, namely make RNA and make protein. The third enigma, which turned out a little bit later, is that if you ask what parts of our DNA are actually active meaning making RNA, but that RNA might not be made into protein, that's about 80% of our DNA. So 1.2% is doing the canonical job, but 80% is doing a transcriptional job. But there are a series of really head-scratching other enigmas. Humans, for example, we have it remarkably hard time making a new baby. Now, let me explain in part why that is or what's going on. So it turns out that we can now look in very, very early fertilized eggs from IVF, but also from prospective mothers who freeze down their eggs, for example. We can look in those and count the number of chromosomes. And it turns out when we do this, somewhere between 40 and 60%, maybe up to 70% of human fertilized eggs die almost instantaneously because they've got the wrong number of chromosomes. I told you humans should have 46 chromosomes, but it turns out quite a lot right at that very early stage don't, and they die almost instantaneously. And we now estimate that for every child that is actually born, another 2 actually died sometime in utero, i.e. never made it. And the mother might not even know she was ever pregnant at all. What's even more enigmatic about this, however, is when we look at fish and frogs, they don't seem to have this issue. They never actually make eggs with the wrong number of chromosomes. But in addition to that, humans are very, very odd. So it turns out that 5% of human pregnancies are affected by a human-specific disease called preeclampsia, which can kill mother and baby. That's remarkably common. And human birth is also extraordinarily difficult. Historically, a mother's chance of dying during childbirth was around 1% and had a lifetime risk of death in childbirth at around 10%. That's, again, incredibly high. Plus, while I told you that for every baby that's actually born, another 2 died in utero, of those that are born, 1 of the 2 typically died before the age of 5 or so historically. So it's a remarkably inefficient process. And part of the reason for that death, but not the only one, is that we have a large burden of what are called rare diseases. Now, rare disease simply means that that disease affects less than 1 in 2,000 of us. But it turns out that 5 to 10% of us have a rare genetic disease. They are not rare in the sense that not many humans have them. Each one is uncommon, but we have a lot of different ones. So none of this, I would say, makes obvious sense. And so it does seem to challenge the march of progress view that we see. Well, for example, if, frogs and fish can get away with making perfect eggs, why do we have so many imperfect ones? Why do we alone have preeclampsia? Why do we have so much DNA bloating? Now, of course, there are 2 potential sorts of answers to this question. One is that actually our genetics is perfect. We just don't yet understand it. And the other is that there's something wrong about our view of evolution as a perfecting process. So, the premise of my book and of this talk is really very straightforward. And it's if we look at these sorts of phenomena, the ones that don't obviously make sense, and that's all I mean by imperfection. It's just they don't make sense. It's by looking at those that we might hope to come to a fuller understanding of our evolution and the evolutionary process. Are we missing some subtle part of selection, or is there something else going on? Now, what I argue also then in the evolution of imperfection is that there have actually been parallel developments in our understanding of the evolutionary process that explain our rotten genetics, and for the most part, we think it is actually rotten. And I would argue that 2 domains are really preeminent. Now, the first of these is the role of chance, and the relationship between the role of chance, how important chance events are, and how big an organism is and how small its population size is. And this comes down to a thing called the nearly neutral theory, and I'll explain what that is quite shortly. The second, I would argue, is there's something funny about mammals, of which we are one, and that's that we have flexible resource allocation, because we allocate resources across a placenta, and this I will show and argue has some interesting knock-on consequences that explain a lot about our inefficient pregnancies. So I would argue that this thing called the nearly neutral theory was developed by this woman, Tomoko Ota. It's actually the most important development in our understanding of the evolutionary process that's developed over the last 50 years or so. It's very different from Darwin's view of evolution. So let's look at what it actually says. Well, what it says is that actually when you think about evolution, all evolution really is mutations in DNA. So the DNA is slightly changed. That's a mutation. We all have them. We're all born with about 50 to 100 new mutations. And all evolution is those mutations changing frequency in the population. And if you ask, why is this species different from that species? All of those differences must have started out as mutations that were initially rare and became common. So the question is, how do you go from rare to common? What are the processes that could mean a mutation that was just in me when I was born, for example, but we forward project many millions of years or hundreds of thousands of years, becomes more and more common? And Darwinian selection is one of those, but there are alternatives, and the nearly neutral poses a very important alternative. And really what it says is that actually you can think of these mutations as being a bit like slightly dense dust particles, in the sense that they might be slightly harmful to you, but if they're slightly harmful to you, will selection necessarily get rid of them? And what Oter argued, well, actually, like slightly dense dust particles, which should on average go down in frequency, so they start near the floor, gravity should take them down to the carpet. Mutations are the same. They start very, very rare, but by chance can increase or decrease in frequency. So if they're slightly dense, if they're slightly deleterious, they could on the average go down, but they can by chance go up. So this is where the chance goes in. A dust particle should go down, but they can waffle about the room and they can go from near the floor to the ceiling by chance alone, against the force of gravity, or in the mutations case, against the force of selection. And Oter's major contribution then is saying, well, actually what really then matters, so how important is this chance process, it's bobbling about of mutations? They go up in frequency, down in frequency, not because they're good, not because they're bad necessarily, just because of chance, is how tall the ceiling is, which in biological terms, how big is the population? If it's a really big population, that's a lot of lucky breaks that you need to get from floor to ceiling. If it's a small population, not so much. And so what she predicts is that if the population is small, these chance events can dominate. And in dominating, what that's saying is the chance events can start to swamp selection's effects. So selection should knock out harmful mutations. In a small population, that may be a less efficient process. In a large population, it's a very efficient process. The dust particles will eventually settle on the floor because gravity will take them there. Harmful mutations will all eventually be taken out of the population because selection will take them there. But in a small population, some can escape that. So remember that, because this is key. If you've got a small population, and we ancestrally did, selection should be less efficient at getting rid of slightly harmful things. So what does that actually lead us to expect? Well, it does lead us to expect things about genomes. Why? Just imagine there's a mutation. All it is a slight gain of a bit of DNA. Now, that slight gain of a bit of DNA, like a transposable, like a jumping gene, is going to be slightly harmful, probably. There's a little bit more DNA you have to copy, and so on and so forth. So what's going to happen to that small insert? Well, what this theory says is that if you're something like yeast, you have a massive population just because you're tiny. There'll be billions of you. So selection should be efficient. This new mutation is going to slightly harm you. Selection will get rid of it. You end up with a compact line genome. For us, small population sizes, because we're big-bodied things, at least historically we had small populations, selection will be inefficient, and so chance is going to dominate, and these things can just float about, and some will indeed, just by chance, increase in frequency until more or less we all have them. That's so-called drift. So that's the nearly neutral process. So we should end up with a large bloated genome. And so it makes a very nice prediction that if we look at how much genome bloating organisms have, it should be predicted by what their population size is. And it turns out that this is exactly true. So here we have two different measures of genome bloating. One is the bits in our genes that we appear not to need, introns. So how big are they? And the amount of DNA between our genes. And what you see here in both cases is that when the population size is small, you have lots of it. When the population size is big, you have little of it. So here's a good reason to suppose that Oter's onto something, that this nearly neutral process saying it's the population size that matters is actually onto something. And there you can see us in the top left-hand corner, the most bloated in this regard. Now, interestingly, the same theory can be extended and say, actually, if you are a large-bodied organism with a small population size, you have inefficient selection. It also means you're less good at keeping down the mutation rate. Now, we think the mutation rate is always under selection to be as low as is feasibly possible. But at some point, selection stops being efficient in doing that. And so the same model makes an interesting prediction. It says that if you've got very few individuals in your population, selection is less good at keeping the mutation rate down. You should have a higher mutation rate. And it turns out that this is also true. So here again is on the bottom axis, how many individuals are in a species. And on the other axis, the vertical axis, is what is your mutation rate. And as you can see there, we've actually got a very high mutation rate, something like 10 to the minus 8 per base pair per generation. Go down to bacteria and what have you, and there's per base pair is about 10 to the minus 10 or so. And there's one possible one at 10 to the minus 11. Incredibly slow rates of mutation. But as you can see, the general trend seems to be pretty robust. Organisms that have big populations, because they're small, have very low mutation rates. Things with very small populations have high mutation rates. And this explains the first of our many enigmas. Why do we have so many genetic diseases? Well, in no small part we have lots of genetic diseases because we've got a very high mutation rate, generating versions of our genes that have mutations in them. So if we follow this logic further, what it actually says is that while we've got a very large genome, 3 billion base pairs in any one of those 23 chromosomes, a lot of it is likely to be non-functional. It is simply stuff that is accumulated over time that we can't get rid of. Is that true? Well, we can look at fingerprints of selection within our DNA, and the best evidence we have says about 10%, probably not even that, but it's probably about 10% of our DNA that is doing something. So that's the 1.2% that is making protein. There's some other stuff that makes RNAs that are important, and some other stuff that control all of that. Add all of that up and you get something like 8 to 10% of our DNA that appears to be doing something important for us. Most of the rest shows no signs of doing anything important for us. So in this view of DNA then, much of our DNA is what we call junk. It's stuff that you just put away in the loft or in the garage. You can't get rid of it. It just accumulates over time. And that's because we are descended from populations with very few individuals. And when I say we're descended from very few individuals, we think that the human population size was about 10,000 or so when we were doing most of our evolution. And by contrast, there are more bacteria currently sitting in your gut, about 100 trillion, than humans that have ever lived by quite some way. So yes, there's a couple of billion of us now. Historically there weren't, and those small things will always predominate. So what about these funny problems of pregnancy and this resource flexibility problem? Now let me explain that resource flexibility problem in a little bit more detail. So I'd like to turn to a real extreme here that doesn't have resource flexibility, and it's this bird. This is the kiwi. What is remarkable about a kiwi, you can see the kiwi's egg there, is that a kiwi female will invest about 1/4 of her body mass into that one egg. That is a huge amount of investment. Now, what's important about this is not simply the amount that she invests into that egg. It's that all of that investment is done up front. So if the embryo is inviable, for example, she will still have invested all of that body mass into that egg. Turns out they will also sit on the egg, so they waste energy and time if the embryo happens to be inviable. And so we can say if the egg is inviable, there's no compensation for that. She has simply lost all of those resources. She's lost the time and she's lost the energy. There is no compensation coming back from the death of the embryo. Now that's not true in mammals. A key difference here is that initial human embryo is one cell and it's tiny, and the mother hasn't invested up front. So it's a minuscule proportion. So if that embryo dies really, really early, the mother hasn't given away all the resources and she hasn't given away the time. She's not a kiwi. The consequence of that is if that embryo then dies, the mother has saved the resources and saved the time, and that's what we call reproductive compensation. So the death of one embryo leads to a saving that the mother can invest either time-wise or energy-wise into another embryo. And we can actually put some numbers on this. So we can estimate, for example, when we look at singleton births, so a mother has only one kid, versus a twin birth, so a mother has either 2 identical or 2 non-identical kids in the same birth. Turns out twins tend to be a bit smaller, about 40% smaller. Now, being 40% smaller, we can also work out from old plots, turns out that has an effect whereby if one of the 2 die and the surviving kid more or less gets all the resources because the death happened very early on, that surviving kid now has a 10% increased chance of surviving. So it's quite a big effect. That reallocation from the death of one of the 2 twins gives a 10% boost to the survivor. Notice none of that occurred in the kiwi. Now you might think, and you wouldn't be far wrong, that at first sight this looks like a rather perfect system. You save the resources that would have been wasted. That looks like a most effective solution to the problem of reproduction. But it turns out that this does funny things to the evolutionary process. First, what it does is it actually increases the frequency of mutations that kill embryos. And this is because the ones that receive the released resources can also have the lethal mutation if it's what we call recessive, so if it's hidden. So the embryos that have got the two versions of the lethal mutation, they die. Who gets the resources? It's the survivors, but they will have one copy of that lethal mutation. And it turns out that keeps them in populations. And that might explain, we think, part of the reason why 15 to 20%, going up to 70% in mothers over 40, but 15 to 20% of recognised pregnancies as post-week 6 of pregnancy, you've determined you are pregnant, will end in a miscarriage before week 20, usually about week 12 or so. And we think this is part of that explanation. And it also explains, we think, why mammals do, but frogs and amphibians don't, have a problem with embryos that have got many more chromosomes. So here you see a chromosome squash of one of the fortunate ones that did actually survive. 3 copies of chromosome 21 is Down syndrome. Most Down syndrome embryos die in utero, but a few make it through to survive. But in utero, so if we go and look at those very early embryos, you'll find embryos that have got 3 copies or 1 copy of all of these chromosomes typically. 21 is just quite small and they manage to survive some of the time. Now, why do we think it's something to do with degree of reproductive compensation? Well, there appears to be this sort of rather general rule that if you ask what is the rate at which mammals have embryos that have got the wrong number of chromosomes, it turns out it's approximately 1% times the number of chromosomes. So pigs have 18 chromosomes and about 18% of pig embryos have the wrong number of chromosomes. They usually die as well. In mice have 20 chromosomes and their number is 20%. Cows have 30 chromosomes and they have a higher rate of having the wrong number of chromosomes, that's about 30%. And us, we're the slight anomaly. We've got 23 chromosomes, so our rate should be 23%. And actually that's about what you see in young mothers, but we're also deferring reproduction to quite late, and there's a big maternal age effect here. By contrast, in birds, we expect a very small rate because when the chicks develop, they could be competing amongst each other, but before that, in the egg, no. And the rate is 0, as I said, in frogs and fish, where we don't think there's any competition between the offspring, so no possibility of reproductive compensation whatsoever. I'll not go into the precise logic for that, it's quite involved, but do have a look in the book if you want an explanation of exactly why we think reproductive compensation matters. But in no small part, it reduces the costs of having the wrong number of chromosomes because you get some of it back, as it were, from the death of the embryo. Why then this funny thing, preeclampsia? Now, preeclampsia, I do think, is one of the most fascinating disorders of humans. So as I said, it's a human-specific condition of pregnancy, can strike any time week 20 onwards, and it's associated with very high maternal blood pressure, and it's caused by products released by the placenta, and that we know. So often you see in textbooks that placenta is there to take resources from mum into the embryo and they diffuse across, but the placenta also puts things from the baby into the maternal blood system. Now, it can be lethal and the only treatment is an emergency delivery. And the great enigma is it affects a remarkably large number of pregnancies, 5% of all pregnancies. So what's going on? Why is it human specific? And here's what I think is probably the best explanation at the moment. And it comes down to another peculiarity of human pregnancy. So what's peculiar about humans? Well, we do have very large brains. And brains need a lot of feeding in utero. So in mum's tum, we need a lot of food. We have, as a consequence, a really invasive placenta, and that's to maximize surface area and all that. So get as much resources in from mum as is feasibly possible. But it comes with a disadvantage. The mother, you see, should not be seeing the proteins being expressed by the baby. Why? Because she doesn't know about the ones from dad. So this gives you a problem. If the mother does ever get to see those proteins from a distressed placenta or something like that, then the mother can immunologically attack her own fetus because she doesn't know about the proteins coming from dad. How could she? So That is what we think is going on. It's a mother responding to her own fetus, the baby's placenta, so the placenta is from the baby, and immunologically attacking it because she doesn't recognize it. And that's a bit like if you have a viral infection or bacterial infection, your immune system attacks. That goes, it's foreign, I don't know about it, attack. Now, there are ways around this. We don't, for the most part, attack our own proteins. If you do, that's called autoimmunity. We don't typically attack our own gut flora, for example. That's nicely walled off. If you do, that's ulcerative colitis and Crohn's disease. Looks as though it's in no small part what's going on there. But the mother doesn't want to immunologically attack sperm, and she doesn't want to immunologically attack her own fetus. How could she avoid that? Well, this is what we think is going on. We think human intercourse is actually maternal immune priming. That's to say, every time a mother is having unprotected sex and being exposed to semen, that semen actually contains lots of proteins that she will see subsequently in pregnancy at the lining where the placenta meets the mother. So, for example, right at that lining is a protein called sperm-specific antigen number 4. It's not sperm-specific, it's in the sperm. And what we think is things like that are there to train the mother's immune system. So if you have lots of unprotected sex, the mother's immune system is getting trained, and she'll go, oh no, I've seen those proteins before. That's all right, chill. So when you next see them in the placenta, then all will be well. And some nice evidence for this is that the more unprotected sex you had before getting pregnant, then the lower your chances of indeed having preeclampsia. In fact, it turns out that the highest rates of preeclampsia are in fact in surrogate mothers who neither know maternal antigens or paternal antigens. So yes, I would argue we do have a pretty rotten genetic lot, in part because we descended from things with a small population size and so we're accumulating slightly harmful things like insertions, but also because we've got a strange mode of pregnancy and a very invasive placenta. Can we do anything about this? Well, I would argue yes. If our problems come from our big brains and we think preeclampsia may be a problem of the big brains, then we can do something because those same big brains gives us medicine. And so at the moment, for example, we are on a track whereby we're working out which mutations cause a genetic disease. That can lead to improved diagnostics, and it can indeed lead to improved therapies. And for example, at the moment, a very exciting prospect is so-called gene therapy. We identify the gene that's gone wrong and replace it with one that is right. And that's now been licensed in the UK, for example, for 2 forms of haemophilia, sickle cell disease and beta thalassemia. But that may well be big brains curing genetic problems. You could see it like that. So I'll leave you with one last almost thought, as it were, from Goethe. So if Shakespeare thought we were the paragon of animals, Goethe here, Helga Klein, said, "The man with insights enough to admit his limitations come nearest to perfection, and I'd just inserted genome's limitation into that." Well, thank you ever so much for listening. I've been Lawrence Hurst from the Milner Centre for Evolution at the University of Bath.